Introducing our speaker tonight, Dr. Isabelle Raymond Bouchard. Isabelle got her both bachelor's and master's degrees in cell and molecular biology from Simon Fraser University. And she moved to McGill to do her PhD in microbiology, and she stayed on and is now a postdoc at McGill, also working in microbiology in the same group. And she will tell us about how to find life in our solar system. So.
Sorry? Sure. No, I don't want to hold it. Like that. I think it's going to be a funny hairdo. Yeah. Sort of. Where's the cable? All right. Oh, yeah, these are not, all right, exciting. Okay, can everyone hear me? Yeah, good? All right, great, let's do this. All right, I got to carry this thing. Thank you for coming. I'm excited to be here and talk to you guys today about some of the work that I do. I don't do astronomy, I don't do planetary sciences, I'm not a physicist, so why am I here? Well, I'm actually a microbiologist. I see thumbs up, great. Some of my work is actually in astrobiology, and so I'm going to talk to you guys about that today, astrobiology and the search for life in our solar system and in the universe. So just to start off, I guess, does anybody know what we're looking at? Does anybody know what that image is? Yeah? No? No? So it's actually Enceladus, and this is the Cassini spacecraft, and this is a photo of Enceladus. And we'll find out what these things are a little bit later. So this is a little bit of an outline of what I'm going to talk about today. I'm going to give a little bit of an overview of what astrobiology is. We're going to talk a little bit about what life is, because that might be a question you'd have. What is life, actually? We're going to talk a little bit about the origin and the evolution of life on Earth, the potential for life in our solar system, where might we find life if it's there. We're going to talk a little bit about how we could detect that life, what we call biosignatures or biomarkers. Then we'll talk about some new methods of life detection. More specifically, I'll talk a little bit about the work that we're doing in my lab and in my group to try and create new instruments for life detection. And then we'll also talk about some future astrobiology missions and what is outside of our solar system. Astrobiology is basically the study of the origin, evolution, distribution, and future of life in the universe. It's a very interesting field because it brings together people that are in, like me, microbiology, but it also brings geologists and people that do planetary science and chemists and all these different people that are in these different fields that come together to ask, basically, is there a life somewhere else in the universe? So some of the questions that we look at are things like, how does life begin and evolve? Is there life elsewhere in the universe? And if so, how could we detect it? And what is the future of life on Earth and beyond? So maybe the first question that comes out of this, if we're going to talk about how we're going to find life, is, well, what is actually life? What is life? So if we base it on what we know of Earth, because that's all we've got so far, this is life. This is the basic unit of life. If you've ever taken a biology class, you would have seen this. We consider a cell to be the basic unit of life. This is a sort of a diagram of an animal cell, and so you can see it's this kind of round little ball-shaped thing. It has a bunch of things in the inside. I'm not really going to describe what all of this is, but it's got these different compartments, and they have different roles, and they do different things. You might be familiar with the nucleus. This is where DNA is. That's where chromosomes are. That's where all the genetic information is contained inside of cells. Maybe you've heard of mitochondria before. That's where energy comes from. But so basically, this is a cell. And if you were to take a really high-end, fancy microscope and look at a white blood cell in your body, this is what it would look like. This is a white blood cell that you have right now moving around in your body. So very cool. And so there's a reason why we consider this to be the basic unit of life. And it is because cells can do something that nothing else can do. So at a very basic level, they can metabolize things. They can make energy from food. They can break down substances, and they can make new substances. They can reproduce. They can make little baby cells, make more cells with the same DNA. They can also respond to stimuli. So they can send and receive signals from other cells. They can sense things in their environment. They can communicate with each other, with their environment. And so these are the main reasons why we consider cells to be living organisms. And because cells can do this, they're the sort of the basic unit of life. You can then build more complex organisms by putting more and more cells together, which is what ended up happening with us. So there are a few different types of cells. You have some cells that we sometimes consider to be a little bit less complex. And there are bacteria. This is an example of a bacteria cell. It doesn't have all these compartments inside like you see for plants or animals. It has just, this is a chromosome. This is the DNA that's supercoiled. It's kind of all wrapped in together inside, and it has a membrane and a kind of a capsule around it. And then you've got plants or animal cells that are a little bit more complicated. They've got all these little compartments inside that do a variety of different things. But there is one thing that every single cell has in common, and it is that all living organisms, all cells are made up of at least a single cell that contains these organic molecules. These are carbon-based molecules. We call them carbon-based because they're basically made up of a lot of carbons. This is carbon, carbon, carbon. There's hydrogens here and some oxygen. All these molecules are carbon-based. They're chains of carbons with different things attached to them. So all cells have proteins. They have carbohydrates, they have lipids. Lipids are what make up the cell membrane, so it's what's on the outside of your cell. Lipids are fats, right? So you guys might think fats are these terrible things, but actually always keep in mind that the reason why we have cells is because we have lipids that can contain the information inside the cell. So if it wasn't for fats and lipids, you would not be here today. So keep that in mind the next time. Same thing for carbohydrates. The carbs are so bad. Carbs are bad. Carbs are crucial to a cell. If you didn't have carbs, you would not exist. So always keep that in mind when you think about food. They are at the core of what makes an organism, of what allows an organism to be alive. And finally, we've got nucleic acids, DNA and RNA. You hear a lot about DNA all the time in the news. It's very important. Why is it so important? Why is it so crucial? Well, DNA carries the genetic instructions necessary for the growth, development, function, and reproduction of every single living organism. It doesn't matter if you're a bacteria, an archaea, a yeast, a plant, you have DNA, and the DNA carries those instructions, and it is the same in every single organism. And the way in which this information is brought to life inside of cells is the same in every single living organism. So I wanted to kind of give you a bit of a brief overview of it. So this occurs in every single cell, and this is happening inside of cells. There are some slight differences depending on if you're a bacteria or if you're a human, but the ideas are basically the same. So you have a gene here, which is on DNA, and it carries a unique message. Another gene, gene Y, would have a different code, a different message. And inside of cells, this is transformed into a protein. So first it's transformed into something that we call mRNA, or messenger RNA. It's a little messenger piece of information that carries the code that's here. And then inside our cells, we have these structures here, and they make, based on the code that is here, a protein that is a unique sequence of amino acids. Proteins basically control everything that a cell does. They perform a huge array of functions and roles in living organisms. They control the function of cells, signaling, all sorts of things inside of cells, movement, all different types of things are controlled by proteins. And so that's what DNA does. It basically codes for specific proteins, and these things then do certain things inside of cells. All right, so every cell has this, and this is fundamental to all life. And so now I'm going to do a little bit of an overview of the origin and evolution of life. And I'll start off with this, which is basically how we think of life today. there are three main domains of life. And I just want to highlight that you are like right here with all of the animals on Earth. So you think, great, mammals and all these species that are evolved, but actually most of the Earth, most of the world is dominated by microbes. It's dominated by the organisms right here. You have bacteria on this side here. You have archaea here, and then out of eventually archaea, we've got eukaryotes, and then animals and fungi up here. But really, we are just the very tip of evolution, and most of our world has been populated by these organisms. So at some point in time, at the very beginning, you had the last common universal ancestor, some type of protocell, some type of bacteria or archaea, we don't really know what it was, something between the two that existed. And we don't know exactly what it was, and we don't know when it came to be. There's different theories about the exact timeline and when this Luca, if you will, came to existence. But we have some theories which I'll get to. We do know, however, even though we don't know exactly when life started, that we needed certain things for life. Certain things had to be there to allow life to occur. This is a list, maybe not fully exhaustive, but of the main things that we needed for life to happen on Earth. We need liquid water. This is primordial. It allows for, it's a great substance for chemical reactions to occur and for things to come together and to move together. We need some essential elements, carbon-based life forms, right? So we need some carbon, we need some hydrogen, some nitrogen, some oxygen, these types of elements. We need a source of food or a source of energy, right? You got to eat, you got to metabolize, you got to do stuff. You need an appropriate temperature range. So this is approximate, and it may be that you could have higher or lower, but we base this on what we know in terms of microorganisms on Earth, organisms that can live or survive at the lowest temperatures and the highest temperatures. We think this is approximately the range that you would need to allow organisms to grow. All right, so these are essentially the components, the conditions that are required for life. So when did life actually come to be on Earth? We're not entirely sure. The current evidence, this maybe not so much for the origin of life, for the first sign of life, is about 3.5 billion years ago. So it would have actually happened sometime here. So this was the, if you were watching the questions before this, you found out that Earth was formed about 4.6 billion years ago. And then sometime here, there was simple carbon types of molecules that were first formed. And then around 3.5 billion years ago, right here, actually, this is maybe slightly outdated. So you had maybe Luca, or the first last common ancestor. And then eventually you got prokaryotic cells, photosynthetic cells, oxygen-breathing cells. This is a very, very important part of our evolution because when cyanobacteria, so here when we talk about oxygen-breathing cells, we're talking about a type of microorganism called cyanobacteria, and they release oxygen, and they are primarily responsible for oxygen becoming such a part of our atmosphere and what eventually allowed for the development of higher life forms or more complex life forms like us. So the reason why you're alive today and you're here today is because microbes were there to help you out. Remember that. The next thing you have like strep throat and you're like, oh, bacteria, terrible things. No, they are the reason why you live today. So remember that. They're actually really great organisms, very cool. And very few of them are actually pathogens to you or harmful to you. The vast majority of them are just out there to help you out and just kind of trying to make a life of it just like you are. All right, so eventually we went on. Once we got oxygen, we were able to have more complex types of cells. We got eukaryotic cells, eventually multicellular organisms. So I mentioned that our sort of earliest sign for life was about 3.5 billion years ago. So I wanted to highlight some of this oldest evidence of life, microfossils, which are very interesting. If you listen to the news maybe last year and not that long ago, you might have heard that they had found evidence for the oldest microfossils known to date, dated to 4.3 and 3.8 billion years ago, which is actually quite a bit before what I just said. And this was in northern Quebec. Cool, right? They found these tubular structures that they think are the incrustations or the leftover kind of crust of where a microbe would have been a very, very long time ago. These organisms would have lived in hydrothermal vents in water. This is one of the main theories of where we think life might have originated, which would be in these warm, hot hydrothermal vents where you could have a lot of these chemical reactions going on in water. It's very important to note, however, when you hear these stories, and this is something that maybe when you read this in the news, they don't tell you this, is there are non-biological processes that can create similar structures. So it's important to remember that. This is not actually really conclusive proof of sort of the oldest evidence of life. It's quite possible that it is, but it's also possible that there are geologic processes that cause these formations. So something to keep in mind. About the best evidence we have right now is about 3.5 billion years ago in Western Australia. And you have these filamentous types of structures. You can see kind of a close-up of it here, which appear to be these assemblages of microorganisms. So it'd be one microorganism on top of the other, kind of forming this big chain going up. This was also very debated when it first came out. There are papers published afterwards that were like, no, that's not actually true. That's not what's going on. These are, again, abiotic formations or non-biological formations. And so they had a back and forth, and a few different papers were published with presenting different evidence for and against. But they just, at the end of last year, actually, the group that originally found these structures was able to use carbon signatures. So we human beings use carbon in a way that's different than minerals do or rocks. Rocks have carbon, lots of things have carbon, but we use carbon a little bit differently, and we have specific carbon signatures associated with living organisms. And there are specific signatures associated with bacteria and archaea and stuff like that with regards to how they use carbon. So what they did is they took the carbon signatures of these things and they said, how similar is it to bacteria and archaea that we have today? And they actually found that they were almost exact to bacteria and archaea that we have today in the world in terms of their carbon signatures. This is the strongest case to date that we actually have a real microfossil. So we could say it's quite likely about 3.5 billion years ago we have sort of the first evidence for microbial life. Okay, so I've talked about microbial life, but I did feel like I should maybe tell you what I mean when I talk about microbes. Just in case you weren't quite sure, a microorganism or a microbe is any microscopic organism that must be greatly magnified to be seen. So something very small that you have to use a microscope to see. They are usually single-celled, like bacteria, for example, or archaea. But sometimes you can have more than one cell together, like yeast or fungi. Sometimes you have microscopic animals. This is a tardigrade or a water bear. Maybe some of you have heard of them or seen them. Have you guys heard news stories about tardigrades and how amazing they are? Maybe indestructible organisms. Great, so wonderful. Okay, so I'm here to tell you today that microbes rule the world. They definitely do. They will probably outlive us all. They are amazing organisms that have evolved sophisticated and varied mechanisms to live in a huge range of environments on Earth, including places that we would consider extreme and where we probably wouldn't last more than a second or two. They can live in thermal springs. These are hot, acidic thermal springs. This is at Yellowstone National Park. The orange that you see there is caused by microorganisms. It's carotenoids, it's pigments. It's the same thing that's in carrots. You know, the orange and the carrots. Microbes also make these pigments, okay? So they live in these things. So there have been cases, unfortunately, of humans falling into these springs, and they melt, literally. It's not fun to think about. The microbes here just live in there. Sometimes you also have green microorganisms that have green pigments, and they're just chilling. For them, it's like, yeah, whatever, you know. We have permafrost, which is frozen ground. Microorganisms can live in there. They live in the ground. They live in the ice. They can live inside rocks. The green that you see there are microorganisms that form the little niche, little holes in the rock and are just hanging out and stuff. These are salt ponds in San Francisco, and the color that you see again is caused by microorganisms that are growing in these salt ponds. They like the salt. They just like to hang out there. This is Lost Hammer Spring, which is a spring, a sub-zero saline spring in the Canadian Arctic. Yes, this is me. This looks like snow, doesn't it? It's not snow, it's salt. This is 24% salt. Seawater is maybe 3.5% salt. You have 24% salt. This is a subsurface, so it's an underwater spring that comes to the surface. There's a lot of methane here, all sorts of things, and we find microorganisms in these environments. Microorganisms in ice. I just mentioned tardigrades a second ago. They might be indestructible. Who knows? They can survive the vacuum of space, including dehydration with solar radiation. You can dehydrate them. They lose all their water and they go into little balls like this. And they can survive for a tremendous amount of time in these, and then you just add them back into water and ploop, they just kind of come back to life. You can boil them, you can deep freeze them, you can crush them under pressure 6,000 times the atmosphere. They're okay, they don't care. Yeah, just hanging out, waiting for, all right, so microbes are the best. We've clearly figured that out. They can get their energy and their food from many different types of sources. This is actually very key. One of the reasons that they can grow in these environments is because they can utilize many different types of sources of food and energy. All different types of organic molecules, even more than us. They can eat crude oil. They don't care. Light, rocks, minerals, carbon dioxide, methane. They eat methane. They make methane, whatever, more. They have mechanisms to tolerate higher and cold temperatures more than us. They can tolerate acids more than us, salts, very little nutrients. They can live in environments that have very little food for a long time, lower amounts of water than we can tolerate for sure, some amount of radiation. Many microorganisms don't need oxygen, so they can just do anaerobic respiration. So the main question that comes out of all of this is these guys are hardy, they can live in many different environments and conditions. Could they exist elsewhere in our solar system? Currently, they really are our best candidates to survive some of the extreme conditions in our solar system. So now we're going to talk a little bit about some of the bodies or places in our solar system where we think that life could exist. So these are essentially some of the best candidates for life in our solar system. And I'm really going to focus on 3 today. Mars, partly because we know more about it, and there's some interesting things about it. Europa and Enceladus, because they are so-called ocean worlds, and they have liquid water, probably. As a result of that, the first thing you need for life is liquid water. So for us, they're very interesting. Okay, so let's start with Mars. So this is a selfie of Curiosity rover on Mars. So I'll start off by telling you we don't actually think there's life right at the surface of Mars because it is extremely dry and there's very high radiation, and it is also very cold, and there's not a lot of nutrients. It's not a great environment even for microbes. We think it's rather unlikely they would be right at the surface, but we do think they could be in different places, and so we'll get to that. It's still quite cold, although during the day at the mid portion of Mars, it actually gets kind of nicely warm. Get like a little blanket, get a little bit of sun out there, I don't know. All right, so why we think there might be or could be life on Mars, or could have been life on Mars? Well, there is definitely evidence for the presence of liquid water on Mars in the past. We don't necessarily believe there is liquid water now, but there is evidence, different fluvial channels, different riverbeds, different things that look exactly like places on Earth where we have water, where we've had water, liquid water. So there is a presence of some organic molecules. We haven't discovered that many, but we have found some chlorinated type of carbon molecules, which we know, we just found out earlier that organic carbon molecules are very important for life. We don't know exactly where they come from. They're not necessarily from biological origin, but they are there. And there are a number of known energy sources of food sources for microbes on Mars. I'll show you guys a table a little bit later, and you'll see how many different types there are. So, okay, if microbes are not at the surface, then where might we find microbes? Well, there's this, for example. We believe that there is ice not too far from the surface of Mars. This is a cross-section, so you think of kind of a slope, and it's kind of eroding, and you're able to see what looks to be ice, these thick ice deposits that are not too far down, maybe about 1 to 2 meters below the surface. That's interesting for us as microbiologists because microbes can live on ice in permafrost or in the soil here on Earth. we also sometimes see what we call recurring slope lineae. There are these kind of dark lines that are observed appearing seasonally on the Martian surface. There is some evidence that they could contain hydrated salts and water. They certainly are salts, perchlorates and such, but this is a kind of an ongoing question. Could there be, for a very short amount of time, some water right at the surface? We don't really have enough information to know exactly what's going on there, but it is very interesting to think about. We have microbes on Earth that can live in cold, salty water, so why not? I wanted to introduce one of these organisms to you. This is an organism that we work with in the lab. It is called Planococcus halocriophilus. Halo means salt, cryo means cold, philus means likes cold and salt. This bacteria was isolated from permafrost, which is, again, I said, frozen ground in the Canadian Arctic. I'll show you where a little bit later. It lives in veins, so even though the soil is frozen, the way that we think microbes can survive in the soil is in these thin films or veins of liquid water. So when the water freezes, there's particles and salts that kind of go out and end up in these little thin films around soil particles that are basically salty brines. They're just salty water. And that's where we think these microbes survive in the permafrost. Permafrost temperatures can go down to -17, but the water doesn't freeze because of all that salt that's there. Just like when you put salt outside, right, so that you don't get ice, same idea. In the lab, we've isolated this organism, and we know that it can tolerate up to 19% salt. It can grow down to -15 degrees Celsius, and we know that it's active at -25. It's not reproducing, meaning it's not making other copies of itself or other cells. But it is actively metabolizing, if you will, respiring. So it's still alive, it's just maybe waiting for conditions to get a little bit better. Okay, so we've also got some evidence of maybe methane on Mars. And the question there being, is there any association or link with microbes? We don't really know, and we're still not sure exactly if there is methane or not or what's going on there. But there was some evidence of maybe methane in the atmosphere on Mars. And so where does it come from? Who knows? But I wanted to highlight where microbes could fit into this equation. So this is a big, complicated thing, looking at where methane sources and sinks could be on Mars. We don't really need to know all of that, other than to look at these two things here. So we know that on Earth we have organisms, archaea, they're methanogens, and they make methane. So just like we release CO2, they release methane. So it's interesting to think about whether that could be at role here. We don't necessarily think that's the case, or 100% know that that's the case. It could very well be that there are abiotic processes that are creating the methane. But even if that's the case, even if you have rock or geologic processes that are creating methane, we have organisms on Earth that also eat methane, methanotrophs. They're bacteria, and they eat methane, just like we eat chips or something. So is that something that could be going on there? We don't really know, but it's a possibility. So I wanted to show you guys this table because it really nicely shows you, and I'm not going to go through all of this, you don't have to memorize this or anything, but this is just showing potential energy sources on Mars. And so this is a list of these different types of the different types of minerals that microbes can use on Mars for energy. So that's quite a long list of different things that microbes can use. I'm not saying they're there, but it's definitely very interesting for us as microbiologists, because we definitely think perhaps in the subsurface, maybe in ice, or maybe just in the subsurface, perhaps where the conditions are not as harsh, there's not as much solar radiation, it's not as cold, perhaps there we could find life. And we certainly have components that are energy and food that they would need. All right, so that's enough for Mars. Let's talk about another place. Let's talk about Enceladus. So this is Enceladus. It is a moon of Saturn. This is real, by the way. These are jets, or thought to be jets of liquid water, and we'll get to that in a second. And so it is the 6th largest moon of Saturn. It looks like this. It's a big ice ball. At least it's covered in clean ice. It has a surface temperature of just a nice -198 degrees Celsius. But it does have, what you just saw there, cryovolcanoes that shoot jets of water vapor near the South Pole. It kind of caught everyone by surprise when they saw that. And this might indicate that there's actually liquid water under the ice. Most of this we know from the Cassini spacecraft, which you can see there, which went around Saturn and its moons. And so what kind of data did they get? Well, this is the paper that kind of looked at what was in these water vapor jets that were coming out of Enceladus. This is them here again. And so first, we think that might be because there's a liquid ocean, but we also see all these compounds. We see some methane, water, we see some simple organics, some complex organics, some carbon dioxide. And that's interesting because we know that we can grow microbes with these compounds in the lab. So you have a lot of what you need there for life to potentially exist. So what's happening underground? Why are we getting these jets or these plumes? Maybe there's a warmer, salty subsurface ocean. So this is a kind of a cross-section, and then what it might look like. You've got an ice shell up there, an ocean underneath. You've got these interactions here at the water-rock interface, and you're getting these hydrothermal vents, these really warm hydrothermal vents that are shooting up warm water. So on Earth, we have organisms, archaea, that live in hydrothermal vents. And I told you guys a little earlier that one of the theories where life could have originated on Earth is in hydrothermal vents in water at the 3.5, 3.6 billion years ago. So very interesting to think about. What's going on underneath the ice? What's going on Enceladus? So the last place we're going to talk about is Europa. Europa is a moon of Jupiter. It is also very icy. It's also a big ice ball. It has these really funky kind of striations. It is a moon of Jupiter. I just said that. It is also covered by an ice shell and has similar maybe temperatures to Enceladus, -160 to -220. And then very much the same idea. There is some evidence that there might be a liquid ocean underneath the ice, which would be about 160 to 150 kilometers in depth. So it's got these really weird striations or these fractures on the surface, and it has what looks like maybe mineral deposits. They don't actually know what it is, but it is interesting to think about where this comes from. And so these ice fractures, interestingly enough, are actually caused by, so it orbits kind of strangely around Jupiter. I wouldn't really be able to explain it in a lot of detail, but it has a very strange orbit around Jupiter, which causes tidal stress that actually squeeze and stretch the surface and cause these fractures and these cracks in the ice. Okay, so I just mentioned maybe a salty liquid ocean. So we also see some evidence for water vapor from Europa. And this was data from the Hubble Space Telescope in both 2014 and 2016, where you can see where they suspect that they're actually also seeing these water plumes coming out of Europa. And again, could then we have a warmer, salty ocean underneath that ice that's a bit warmer than -200 degrees Celsius that could harbor life? So an interesting question to think about. Okay, so maybe there's life in these places. If there is life, how could we detect it? So that's a little bit trickier. Well, what would it look like and how can we detect it? So you probably figured out by now, because I've been hammering it for a while, that it probably looks like microorganisms because they could probably tolerate some of these harsher environments. A little too harsh for us to be in, but you never know. Like maybe we'll find a green man walking around one day. But we also kind of, a lot of times assume that life will be carbon-based, and that could be a wrong assumption. It might not be. We don't actually know, but because all the evidence we have on Earth, the only thing we know on Earth is carbon-based life forms. So a lot of when we look at these, we look for signs of carbon-based life. So what we do essentially is look for biosignatures or biomarkers, which is any substance that provides scientific evidence of past or present life. Could be a cell, it could be an actual organism, could be an element, could be an isotope. These are these carbon, I told you that there are carbon signatures in microorganisms or in different living organisms. Could be molecules, could be some phenomenon. So I wanted to give you a little bit of a list of the different things we can look for so you have some idea of what the potential is. So an actual living organism, a living cell, obviously. We could have microfossils like what you guys saw earlier, right? Preserved fossilized parts of cells, or maybe just a mineral cast. Those tubes that you saw, those red tubes, were actually thought to be maybe a mineral cast of what would have been a microorganism. Physical structures, stromatolites or things like that, structures that were created by microorganisms. You could have a mineral structure that's indicative of biological activity. That's a little bit tricky because, again, there's a lot of non-biological processes that cause minerals to, that can look like structures that could be caused by also biological activity. So it's a little bit tricky. You can look for evidence of metabolic activity in atmospheric gases or in carbon signatures. This is cyanobacteria that I told you produce oxygen. You can also, of course, look for the building blocks of life, or carbon-based compounds, organic molecules, amino acids, proteins, lipids, fatty acids, and things like that. There are, however, some limitations currently to our ability to detect biosignatures and to what is actually out there, what we're looking for on these missions. You know, Curiosity is on Mars, but you might be surprised to know it doesn't actually have instruments to detect life. It has instruments to detect the potential, perhaps, for life, but not actually life. So we have some current limitations, and I've mentioned this a couple of times. Some biosignatures can be formed by non-biological processes, so it's always a little bit tricky when we see evidence potentially for life. We have to ask ourselves, how else could this be formed, and try and disprove that to make sure that we're actually looking at life. So always something to think about. So I just said this, but current instruments and rover missions are not that well equipped to detect life. They can detect certain elements necessary for life. Maybe they can detect some amino acids, but these could be formed by other processes also. So not necessarily proof of life, but definitely an indication that you might have the conditions or the molecules necessary for life. And there's a lot, this is important, certainly for Mars, for example, there's no drill for subsurface exploration. That's something that you want. If there's microbes, they're probably in the subsurface, and we need future missions to have drills that can actually look at what's going on underneath the surface. The Viking missions in the 1970s were actually the last space missions to carry equipment for live detection. They did actual biology experiments. So one of the things that comes out of this is that we do need better life detection instruments, and we need astrobiology-focused missions to go into space in the future that are actually focused on astrobiology and looking for life. So one of the things that we are doing in the lab, and there's different groups working on different aspects of it, lots of different groups looking to create life detection instruments that could provide unambiguous proof of life. We focus on a life detection platform that is based at least partly on sequencing and detection of DNA and RNA. There's collaboration with a few different groups there. Part of the reason why we like DNA is because it is an unambiguous sign of life. The only things on Earth that have DNA are living organisms. If you have DNA, you pretty much have a living, or had at some point, a living organism. And it can be readily detected. So we have sort of this microlife platform that we are working to develop, and we're kind of at the beginning of this, so we do have a little ways to go. But our idea is to bring different instruments that already exist, that we use in different fields, and kind of bring them together and use them to create this platform. And so the way that this would work is you would get an environmental sample. So we're working with groups to create a micro drill. You're going to see some images of that in a second. That could go, for example, on a rover. This would be part of a platform that could go on a rover. You could go somewhere, wherever you like, and you could drill into the ground, and you can get some samples from the environment. You could do, we are looking to develop what we call a microbial activity microassay that can detect metabolically active cells, so viable cells, active cells that are metabolizing sources. This is a little plate where you can basically put a dye and you can put nutrients or different substances, and if the organism uses that substance, it turns into a color like this. So you know there is a metabolically active organism in that well, or in that hole. This is an example of a prototype that we're looking to try and create. We are also, I just mentioned nucleic acid sequencing. So we're working with an instrument called the Mrna, and I brought it to show you guys because it's so cool and it's so small and it's great. So normal sequencers, when you talk about sequencing DNA and RNA, like the human genome was sequenced, the thing is like this, right? This is how big sequencers are in like a lab. What we work with is this big. This is a sequencer. This could sequence your DNA, everyone's DNA, not all at once, but it could sequence it. So this is pretty incredible, and it wasn't developed necessarily for astrobiology purposes, but when we saw this, we were like, this is great, because what you want to put on a rover, when you're talking about astrobiology, when you're talking about going to Mars or Europa and Enceladus, you need something that is small, that is light, that is portable, that is robust. It has to fit on a rover with a bunch of different instruments, and the rover is not that big. You can't have something like this this going to Mars. You need something that doesn't use a lot of energy. So something compact, lightweight, robust. This is great for that. So we are working to try and develop this a little bit more and try to integrate it into a larger platform where we could take, this could be incorporated onto a payload or a platform that could go and sequence DNA and RNA, take a sample and see if there's any nucleic acids, and if there are, it would be able to sequence it. We also have in a different portion of it with a different collaborator looking at antibody-based detection of different biomarkers, including cells and amino acids and things like that. Let's see, there we go. I'm going to focus a little bit on this just because that's an area that's the area that I'm more working on. So we'll talk a little bit more about the MinION. You just saw what it looks like. So the way that we would envision this to work is that you have a sample acquisition and a micro drill. Again, this is kind of in development. This is just kind of an image of a prototype, but the actual thing would not quite look like this. That would be able to drill and take a sample in a subsurface or in ice. And then this is an example of a really big core. We're talking about a micro drill, so we're talking about really little cores. But this is a big permafrost core that we got in the Arctic, just a side note. Then you have to take, because this is a soil sample or an ice sample, there's lots of other things there. So you have to remove the soil, remove the other things, and then just isolate DNA and RNA from that sample. And then you can put it into the MinION and you can sequence and you can analyze that data. So obviously, we are working to develop this platform, and we can't just put a MinION on a payload or on a rover and send it to Mars and see if it works. So we have to develop this on Earth. And so we develop this in places that we call analog sites. They are places on Earth that are similar to places that we might find on Mars, Europa, or Enceladus, or other places, if you will. They're harsher, extreme environments, cold, salty, things like that. The place that and there are different places in the world, the place that we go to is called Axel Heiberg Island in the Canadian High Arctic. There is a McGill Arctic Research Station, which stands for Mars, which is great. And I told you guys earlier, Planococcus helicrophilus, right? I talked about that organism. It came from here, Eureka on Ellesmere Island. Anyways, so we go up here, and now I regret I didn't put well, no, yes, there are some. I was like, there's no photo of the site. There are some photos. So we use this place as a bit of a sub-in for Mars, for these places, and there are analog sites there. They're habitats that could be like what we might find in these places. So these are some examples of analog sites. You saw them earlier. This is the famous Lost Hammer Spring with a lot of salt. This is a photo of in winter, April. This is what we, so you guys complain, it's cold here, there's snow. This is in April, this is what it looks like there, so yeah. So we've got permafrost, we've got sub-zero saline springs, we've got ice, again, we've got indoliths, we've got microbial mats. So we've got these extreme sites, and what we do is we go to these places, we take samples from these environments, and we try and do exactly that. These are low biomass samples, they're harsh environments. Can we get DNA out of them? Can we sequence it? Can we test these different instruments? We're going to be going soon in July, and we're going to try and test our first prototype drill. And we've also got different components. I kind of went over it quickly, but we actually have different components and different instruments. We're going to be taking those with us and testing them again this year. So we're trying to optimize the system, but we did a couple years ago, not me, but a previous student in the lab, Jackie Gordio. They went up to the Arctic with the MinION sequencer. I mentioned all of this already, so you know that it's great if you wanted to put it on a rover vehicle, and they did exactly what I just said. They took permafrost samples and were able to get the DNA out, and then they sequenced it, and this was published in Frontiers in Microbiology, and I just wanted to show you a slide so you have some idea of what that looks like. This is the data that you get from it. Try and make sense of all of that, huh? But I just want to focus on this one. Yeah, there's lots of microbes, right? I said microbes are everywhere. Look at how, like, it's insane. There's much diversity in the microbial world, but most of these, I just want to focus on this. Most of these are bacteria. You can see you've got some eukaryotes there, some archaea, and some viruses. So this is what we hope to be able to do one day, perhaps, in space. And so we are, so far, we have been successful in testing these instruments at our Arctic sites, but we've only done it once or a couple of times. that we're still trying to work out some kinks and optimize the system. So we will continue to test and optimize this, different components and instruments, and we do hope to be able to eventually integrate this into an automated platform with some engineers to help, because I am not an engineer. We obviously, this is not going to happen anytime in the near future, but we do hope potentially on a future astrobiology mission we could see a payload like this. And if it would be great if it were to find something, but who knows. Okay, so I did want to highlight some, just towards the end, some future astrobiology missions that have at least something more astrobiology related. So we've got an Exo Mars 2020 rover. This is a kind of diagram of it that is supposed to go in 2020. That will have a surface rover. It's going to have a drill, a little one, I think, not able to go very far, but at least be able to go a little bit into the subsurface on Mars. It will have, I believe, radar to search for subsurface water, potentially, if it's there, which would be very cool. It has some instruments capable of identifying, again, thinking about the orange in the carrots, some pigments, maybe some amino acids, although I did mention earlier that amino acids can also be formed by abiotic processes. So always something to keep in mind. We also have this, I was talking about methane on Mars. Is there methane there? What's going on? There is a trace gas orbiter, which is orbiting right now, and that is supposed to start taking measurements very soon, that will actually look at the presence and potential of methane and other gases and whether it could actually be a biologic origin or abiotic. We've also got, they want to go to Europa, and I think Clipper might be, I think they've actually given a name to this mission now, might be called Clipper. They are hoping to go to Europa and actually look for signs of biosignatures, maybe habitability, just look at the surface and what's going on there. And finally, I'll just end with outside of our solar system. There are some exoplanets, some places outside of our solar system that we think are in a habitable zone. That doesn't mean by any stretch of the imagination that there's anything inhabiting these places, but it is always interesting to think about. We found quite a few of them, and this is an example of where the habitable zone would be compared to, for example, the Kepler system in our solar system. So something to think about for sure, whether or not there could be life in one of these places. And there's more to come. There is, if you guys watched the news yesterday, the day before yesterday, when did it, yes, yeah. So the test is Exoplanet Hunter, I like that name. It's going to look for, survey the sky with very powerful cameras looking for new exoplanets, including maybe some that could harbor life. So that's it. I can't tell you if there's life or not in our solar system or in the universe, but if you ask me if I think there is, yes, I do. And I do hope maybe I'll be alive when we find it, but who knows? Maybe not. Anyways, that's it. Thank you. What is the purpose of my research? You don't want to know if there's life in the universe? So you don't care? Well, that's interesting. I definitely think for me, one of the reasons why, well, I think life is amazing. I'm a microbiologist. I think microbes are amazing, but I think life is amazing. And it's interesting for me to think about where that life comes from, how it got there, how, we don't really understand. I showed you, you saw that we don't know exactly how life started on Earth. So it's very interesting to think about the conditions in these places that caused Earth. How is that different or like her conditions? I think it helps us better understand her own life and the evolution of life, both on Earth and in the universe. So that's one of the main reasons why I'm really interested in it. Plus, it's so cool. But anyways. Yep. So not really as far as I know, but maybe I've missed something. But when we talk about, I talked about biosignatures. So certainly something like where if you had a So where you could have casts of an extinct type of organism. So sometimes you might be able to detect what looks like where an organism could have been, even if it's not carbon-based. You might have some evidence of, because of the way it treats a mineral or it reacts with its environment, you could find some evidence for a living organism. Wouldn't necessarily have to be carbon-based, but we have very little to go on, because if it isn't, we don't know what it is, and we wouldn't know even how to begin to like look for it, if that makes sense. Yeah. Yeah. Yeah, for sure. Yes, yeah. So for sure. So for example, with the was it the Vikings? I can't remember. It might have been the Viking land. So there has been incidents where they thought they detected something, and they were like, I think this might actually just be a contaminant. So yes, so you have to take lots of precautions, and yes, it's pretty much impossible to fully sterilize anything. So whatever is going to Mars is going to probably, or wherever, is going to carry something on its surface. One of the ways that we can differentiate them is because we know we have a good idea of life on Earth. So if we find something that looks exactly like us, it's got the same exact signs as like E. coli, well, it's probably E. coli, right? And we probably just contaminated the surface. So we do expect at a DNA level, at a molecular level, at a metabolic level, some differences in these organisms, obviously, that are, if we think of the diversity on Earth, we probably think this is going to be a little bit different if we do find something. So we can compare it to what we know on Earth and see how close it is to what we, and if it's anything like what is on Earth. So that's one way. How to prevent it? There's all sorts of things you can do with UV and with cleaning the surfaces, but it's pretty much impossible to 100%, have nothing zilch going out there. That's also planetary protection is a whole other thing that we won't get into maybe, but yeah. Yeah, so the drill on the little drill, for example, on the Exo Mars is going to Mars, and that one is not going to be able to go very far under the subsurface. Eventually, you need a fairly big drill. What's interesting about Enceladus, so when I said micro drill, I meant more that the drill itself, the diameter is small, but you could have like a long drill if you want to. But it's the bigger drill you have, the more energy, the harder it is to design and to. So even going to Europa or something like that, I don't envision that they would ever try to get to the ocean. They would just try and go a little bit further into the ground and see what they get down there. So for what's happening with Mars right now, that's what it is. They're only going to go, a few centimeters, and they're going to see what's there. So we're a ways away from being able to drill into the ocean. Yeah. Yeah, so that's one of the theories, that on meteors, we know that there's organic compounds. We know there's amino acids. It's entirely possible. So, one of the criticisms sometimes people tell us about our work is we're focusing on DNA and RNA. And again, maybe a life form isn't based on DNA or RNA. But we know that amino acids and carbon-based molecules are on meteors. So if a meteor came to Earth and brought these with it, it's entirely possible it went to Mars and brought some of those things with it also, right? So it's definitely a possibility. We don't really know right now. Yes. 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 if I could call NASA and be like, hey, dudes, like, yeah, let's get the ball rolling. So yeah, a lot of these decisions are made higher up at the, maybe even bureaucratic level. And, there's a lot of scientists at NASA and they try to prioritize. And it's true that it is important. If we want to talk about life, we do have to understand the geology of these places and we have to understand how all of that is working. So it is important. There's a whole issue, which I didn't talk about, of, again, planetary protection. So there's a lot of concern that if we go on Mars or these places looking for life, or a place where we think there could be life, we will either contaminate it or mess it up somehow. And so even though we know there are places where, there's places on Mars that might be good candidates for where we could find life, we're not actually going to these places because we're concerned about planetary protection. So there's a whole debate on a scientific bureaucratic level about where to go, and do we go to these places or not, and when do we go to these places, and how do we go to these places. But of course, if Elon Musk sends, as soon as a human lands on Mars, it's like, pff, kaput, because we are living like microbial, whatever, incubators, right? And so we would have to try and get to these places before we actually send humans to Mars. But yeah. Yes, so you're talking about the Mrna? Yeah, for sure. It hasn't been around for that long, it's just a few years. The company is constantly, so that's one thing we're having to deal with, is that originally, I would be honest with you and say it wasn't that great, but it has come a long way in the last few years, and they're constantly developing better technologies. And so we are trying to work with that and kind of like move forward as they do. Yes. They're okay, for sure. We've actually been able to, so I don't know how much you know about DNA extractions or stuff like that, but we usually have to do different steps. So we have to do extraction of the DNA, then we have to do a library preparation to prepare the DNA for sequencing. We call it a library, but just means different DNA molecules. We're preparing it to sequence. And we've been able to do that even with samples that haven't been purified very much. So even fairly crude samples, we've been able to get some sequencing. So it'll work even if your sample isn't that great. It does have a higher error rate, meaning that sometimes there could be an error in the base. So the DNAs are made of bases, but it could be an error when it's sequencing. It's not that high, but it is higher than other types of sequencing methods like Illumina, for example. And that is something they're actively working on. But we are definitely very confident when we get sequences that we're getting DNA sequences from our samples.
So we're out of time. If you have any more questions, Isabelle will stick around for a few more minutes if you want to come on down and ask her. But thank you for coming, and let's thank Isabelle.
Thank you.